In this video, you will learn about the lymphatic and immune system, including its functions, structures, and how it protects the body. First, I will list the major functions of the lymphatic and immune system. Then, I will describe how lymph is formed and transported through lymphatic vessels, lymph nodes, and lymphatic ducts. I will describe the structure, location, and function of lymphatic organs, lymph nodes, spleen, tonsils, Peyer's patches, thymus, and bone marrow. Then I will contrast the functions of primary lymphoid organs with the functions of secondary lymphoid organs. Finally, I will distinguish between innate and adaptive immunity. The lymphatic system has three major functions. First, it regulates interstitial fluid volume. Fluid constantly leaks out of blood capillaries due to blood pressure. This fluid becomes extracellular fluid surrounding the cells. If this fluid is not returned to the bloodstream, it would accumulate in tissues. Lymphatic vessels collect this excess fluid. Once the fluid enters lymphatic vessels, it is called lymph. The lymphatic system then returns this fluid to the bloodstream, helping maintain blood volume. The second major function is immunity. The immune system protects the body from pathogens such as bacteria and viruses. Leukocytes play a key role in this defense. These cells travel through tissues and attack harmful organisms. Lymphoid organs contain large numbers of leukocytes, including lymphocytes. B and T cells are the lymphocytes responsible for adaptive immunity. Lymph nodes are important lymphoid organs that filter lymph and remove pathogens. The third function is the absorption of dietary fats. In the small intestine, specialized lymphatic vessels called lacteals absorb lipids. These fats enter the lymphatic system before eventually reaching the bloodstream. Next, let's follow the path of lymph through the body. Lymph begins in lymphatic capillaries. These are the smallest lymphatic vessels and are found near blood capillaries. They have one-way valves that allow fluid to enter but not leave. As interstitial pressure increases, fluid moves into these capillaries. Lymph then travels through larger lymphatic vessels. These vessels contain valves, similar to veins, to keep lymph moving in one direction. They carry lymph toward lymph nodes. Lymph nodes filter the lymph. They remove debris, pathogens, and abnormal cells. Eventually, lymph enters lymphatic ducts, which return it to the bloodstream. The right lymphatic duct drains the upper right portion of the body into the right subclavian vein. The thoracic duct drains most of the rest of the body into the left subclavian vein. If lymphatic drainage is disrupted, fluid can accumulate. Edema is swelling caused by excess interstitial fluid. Lymphedema occurs when lymphatic vessels are damaged or blocked. Now let's discuss lymphoid organs. These organs contain clusters of leukocytes and are important for immune function. Red bone marrow is lymphoid tissue found in the trabecular cavities of spongy bone and the medullary cavities of long bones. In addition to its essential role in hematopoiesis, this tissue is where B cells mature. Here we see a diagram showing the process of hematopoiesis that occurs in red bone marrow, producing all the formed elements of blood. Myeloid stem cells are the precursors for erythrocytes, platelets, all the granular leukocytes, and monocytes. Lymphoid stem cells are the precursors of all lymphocytes. In red bone marrow, the myeloid and lymphoid precursor cells are arranged in cords surrounding highly permeable capillaries called sinusoids. The large megakaryocytes that produce platelets are a distinctive feature of red bone marrow. A few adipocytes are normally present in red bone marrow. However, the cords of myeloid and lymphoid precursors are replaced with adipose tissue, as red bone marrow is converted to yellow bone marrow. The thymus is where T cells mature. Immature T lymphocytes, called thymocytes, migrate from red bone marrow to the thymus. Lymphoid tissue in the thymus is organized into lobules. Each lobule has a layer of cortex surrounding a medulla. Thymic bodies are the most distinctive histological feature of the thymus. Thymic bodies are concentric, layered aggregates of keratinized epithelial cells in the medulla. Lymph nodes filter lymph and contain regions where lymphocytes become activated. The surface of a lymph node is a capsule of fibrous connective tissue surrounding the cortex. Projections of connective tissue that fold deep into the lymph node are called trabeculae. Lymph flows into the lymph node through afferent vessels and travels through the subcapsular and trabecular sinuses in the cortex. As lymph travels through the cortex, it passes by lymphocytes that are organized in spherical clusters called lymphatic follicles. Lymphocytes that become activated divide, forming germinal centers within the lymphoid follicles. Lymph flows from the cortex deeper through medullary sinuses. Effector B lymphocytes, called plasma cells, migrate from germinal centers into the medulla of the lymph node to form linear bundles called cords that surround medullary sinuses and secrete antibodies. Finally, lymph flows out of the lymph node from the medullary sinuses into efferent lymphatic vessels. The histology of the lymph node is characterized by numerous lymphocytes forming lymphoid follicles within the cortex, deep to the capsule, and deep linear bundles of plasma cells forming medullary cords. The spleen is the largest lymphoid organ. It is in the left hypochondriac region, and its primary function is to filter blood. The spleen removes old red blood cells and pathogens. White pulp contains lymphocytes that respond to pathogens. Red pulp contains macrophages that remove worn-out erythrocytes. Blood flows from arterioles into the sinusoidal capillaries of white pulp and then drains from the red pulp into venules. The histology of white pulp is characterized by the high density of lymphocytes organized around arterioles. Red pulp can be distinguished by the lighter staining pattern with a lower density of leukocytes and a relatively high number of erythrocytes. MALT is mucosa-associated lymphoid tissue found in mucous membranes. It filters mucus to protect areas exposed to the external environment. The largest examples are the tonsils that form a protective ring around the pharynx. The pharyngeal tonsil, or adenoid, is located in the nasopharynx. A pair of palatine tonsils are found in the oropharynx, posterior to the soft palate. The lingual tonsil is located at the base of the tongue. The histology of the tonsils is characterized by numerous lymphocytes deep to a stratified squamous epithelium that folds inward to form crypts. Round clusters of lymphocytes, called follicles, or aggregated lymphoid nodules. Within these follicles, actively dividing lymphocytes produce germinal centers. Peyer's patches are mucosa-associated lymphoid tissue that forms a distinctive histological feature of the submucosal layer in the ileum of the small intestine. Lymphocytes in Peyer's patches monitor the mucus of the small intestine to help regulate the bacterial composition of the gastrointestinal tract and respond to pathogens. Now let's compare primary and secondary lymphoid organs. Primary lymphoid organs are where lymphocytes become immunocompetent. This means they are capable of responding to antigens. An antigen is any substance that can bind to lymphocyte receptors. Red bone marrow and the thymus are the primary lymphoid organs. Secondary lymphoid organs are where lymphocytes encounter antigens and become activated. These include lymph nodes, the spleen, and mucosa-associated lymphoid tissues, such as the tonsils and Peyer's patches. Finally, let's distinguish between innate and adaptive immunity. Innate immunity is a rapid, nonspecific defense present at birth. It includes physical barriers, immune cells, and defense proteins. Adaptive immunity is specific to particular pathogens. It develops after exposure to an antigen. A key feature of adaptive immunity is immunological memory. On a second exposure to the same antigen, the response is faster and stronger. Memory B cells and T cells provide this long-term protection. In summary, the lymphatic and immune systems work together to maintain fluid balance, absorb lipids, and protect the body from disease.